This is Chapter 5, Part 4. So we're moving on to persistent infections. Some cells maintain a carrier relationship, is when a cell harbors the virus and is not immediately lysed. This can last from a few weeks to the remainder of the host's life. The virus can remain latent in the cytoplasm. So a provirus is when viral DNA incorporates itself into the DNA of the host cell and is then copied with future generations of the cell. And an example of that is virus. There's others, but I just threw HIV in there as an example. A chronic latent state is when the virus can actually periodically become activated under the influence of various stimuli. A lot of times it's stress. Herpes simplex and herpes zoster are examples of that. Specifically, herpes zoster is chickenpox, and later on in life, if you had chickenpox, you can end up with shingles. So my mantra is always kind of like, if you can avoid it, please do. Getting vaccinated for certain viruses is key. Personally, I have suffered from shingles because I had chickenpox when I was a kid, and you think, oh, shingles is an old person's infection, but it's not really. I've heard of young people getting it in their teens, so it's ultimately brought on by stress and can kind of debilitate you for a while and cause long-term nerve damage. And again, we're talking about chickenpox and then later on in life, shingles. So this is, generally speaking, I try to avoid, any virus out there because nobody really wants to be sick. So when people start, telling me that, well, you gotta make your immune system strong, and that's true to an extent, but I definitely did not enjoy shingles, and in fact, I don't know anybody that has ever had shingles, and I don't think that any of them enjoyed it. So I just do my best to avoid, viruses, especially viruses that you don't necessarily know about, like novel viruses that are rearing their ugly head right now. So just do your best to keep yourself safe is all you can do. So moving on, viruses and cancer. So that's the other thing, is that there are viruses that cause cancer, and experts estimate right now that there's 13% of cancers that are caused by viruses. And that's not just a number they pull out of the sky. It's based on what they've learned so far. They're thinking about 13%. I personally think that number is much higher, but that's just my personal opinion. So transformation is the effect of an oncogenic or cancer-causing virus. Some viruses carry genes that directly cause cancer. Other viruses produce proteins that induce a loss of growth regulation, leading to cancer. Because cancer cells ultimately are, out of control cell growth is what they are. And that's what that means, is that the virus can induce that growth regulation, and then that can lead to tumors. So things like mono, Epstein-Barr, are linked to cancer later on, certain types of lymphoma. Let's see, there's another one, drawing a blank. It'll come to me in a minute. I don't want to hang on here. Human papilloma, HPV, causes cancer. Human T-cell leukemia virus, HTLV1, type 1, that causes cancer. Those are all viruses, and later on can cause cancer. So transformation of cells, increased rate of growth, changes in their chromosomes, changes in the cell surface molecules, and capacity to divide indefinitely. So those are things that viruses can do to cells in the transformation of cells. There's just some interesting information here. I'm not going to read over that. You can review that yourself. Viruses that infect bacteria. So I could stop here, but I think I'm going to keep going because I'm only 5 minutes in here. So viruses that infect bacteria are called bacteriophages or bacteriophages. That means basically bacteria eating. Most contain double-stranded DNA, but some are RNA types that exist as well. Every bacterial species can be parasitized by various specific bacteriophages. The bacteria they infect are often more pathogenic for humans. What that means, what this statement means is if a virus infects a bacteria, it makes that bacteria basically, I have it later on in here, like it gives it superpowers. So it makes it stronger. They're more pathogenic. They're more disease causing once they've been infected with this virus. And this is like, and let me just point out, this is like an uncontrolled situation here, like in vivo. So Teven bacteriophage is the type of virus that infects E. coli. It's got this icosahedral capsid shape. The central tube surrounded by a sheath. There's a collar, a base plate, tail pins, and fibers. And it's like the picture from earlier. It looks like a scary spider. But you can see it punctures. This is supposed to be the bacterial cell wall. It punctures the cell wall and then basically expresses or inserts, or however you want to, the terminology, the DNA into the cell, or injects it. That's the word I was looking for. It injects the nucleic acid into the cell. So two different things we're going to talk about here in this, and we're going to kind of revisit it in the next slide or two. Events in the lytic cycle of a Teven bacteriophage. So this is the life cycle, if you will, or the reproduction cycle of these types of viruses, which are bacteriophages, right? When we say bacteriophage, we're talking about a virus. So we have an E. coli, which is the host. Okay, so this guy gets a hold of the E. coli and adsorption happens. So it attaches to the surface. That's what adsorption is. It's attaching to the surface. Penetration happens, so it punctures it, and then sends it during penetration. The genetic material is injected into the bacterial cell. Okay, here's the existing bacterial DNA. Okay, so in the lysogenic state, when this happens, there's two states here. There's lysogenic and there's lytic. Okay, I'm not going to get into the whole lytic cycle first. We're going to jump over here to the lysogenic stage. So we have adsorption, penetration, and then basically that viral information gets incorporated into the bacterial DNA. That's that little blue thing there in the bacterial DNA. And it's just kind of blown up. So it fuses with it. It becomes part of it. And in the lysogenic state, it becomes kind of a latent state. This is kind of the more dangerous state because this can then multiply by binary fission. So this cell becomes 2, and then those 2 cells become 2 and 2 and 2 and 2. And then all of a sudden we have all these cells with this viral information in it. And at any given time, that cell can revert back to the lytic cycle where that information is copied from that from the genetic material, and all the parts are made following those directions. So then we have all these parts inside of the cell, duplication of the phage components, replication of the viral genetic material, and then we go on to the assembly. So now the viruses are assembled and they mature and they fill up the cell and the cell bursts, right? It lyses. That's what lysis means. It's when the cell bursts. And now we have all these bacteriophages floating around. Now this state, this lysogenic state, can go on for a very long time. I think down here it's inserted, I say the viral DNA molecule is inserted at a specific sites in the bacterial chromosome. The viral DNA is duplicated along with the regular genome and can provide adaptive genes to the host bacterium. So it makes the host smarter, more ability to withstand things, and it replicates and replicates and replicates and replicates, and then later on down the line, it can revert back to the lytic cycle and then come into this third phase of the cycle and make its way through. So that is the life cycle of, or the lytic life cycle of Teven bacteriophages. Remember, bacteriophages are viruses that infect bacteria. So more on this. Lysogeny is the silent viral infection. The temperate phage undergoes adsorption and penetration, and they don't undergo replication or release immediately. That viral DNA enters in an inactive prophage state, inserted into the bacterial chromosome, copied during normal cell division, and then it's referred to as lysogeny. Okay, just like up there, it's a condition in which the host chromosome carries bacteriophage DNA, and then something induces that prophage into the lysogenic cycle. It becomes, in a lysogenic cell, becomes active and progresses through the viral replication and lytic cycle. So I don't know if this cycle can stay like this for, years, hundreds of years. I don't know. I don't know what the process is here. So I don't know exactly what will cause it to revert back to the lytic cycle, but it is two ways that the replication can happen. So the role of lysogeny in human disease. Okay, so remember, we're talking about E. coli here, right? Viruses and E. coli. So how does that affect us? So occasionally, the viral genes in the bacterial chromosome cause the production of toxins or enzymes that the bacterium would not usually have, like giving the virus, this is supposed to say bacteria, like by giving the bacteria superpowers, right? The virus gives the bacteria superpowers, we'll say. During lysogenic conversion is when the bacterium acquires a new trait from that viral phage. Okay, and examples are here about the toxins that are, able to be made. So it says put the phases of the life cycle of animal viruses. Okay, we're reverting back to the first ones we were talking about, not this bacteriophage one, in the correct order, and I put them in order: adsorption, penetration, uncoating, synthesis, assembly, and release. I put them in order for you. It's just a kind of a review. All right, so we're going to stop there, and we'll pick up on hopefully the last part of Chapter 5.